Today we're diving into a piece of technology that is, quite literally, a molecular photocopier. It's a tool so powerful it can find one single genetic needle in a haystack and then print a billion copies of it.
So, let's start with a puzzle. How in the world do you study the DNA you get from just a single drop of blood or a tiny fragment of a fossilized bone? I mean, when you have an amount that's practically invisible, where do you even begin? That's the core problem we're going to solve today.
For a long, long time, the simple answer to that question was you couldn't. The amount of DNA scientists had to work with was just too small. It was like trying to find a single specific needle in a haystack the size of a mountain. The DNA was there, but it was basically invisible and impossible to study.
And think about what that means. For a crime scene investigation, a critical medical test, or trying to understand ancient life, the sample you get is almost always tiny. Without some way to make more of that DNA, all of its secrets, all of its stories would just stay locked away forever.
But then, in the 1980s, an absolutely brilliant solution came along, a technique that could find that one specific needle in the haystack and, get this, make millions and millions of copies. So let's meet our hero, the molecular photocopier. Now, its official name is the polymerase chain reaction, or what everybody just calls PCR. And at its heart, it's a way to take a tiny specific piece of DNA and just amplify it like crazy, creating billions of perfect copies. That photocopier analogy is dead on. It's like finding one specific sentence in a giant library and printing a billion copies of just that one sentence. And listen, this wasn't just some minor step forward in the lab. Oh no. This was a full-blown revolution. It was so important, so world-changing, that its inventor, Kary Mullis, won the Nobel Prize for it in 1993. It completely rewrote the rules of what was possible in biology.
Okay, so how does this miracle machine actually work? Well, like any good recipe, it all comes down to the key ingredients. Let's take a look at what you need to make billions of DNA copies from, well, basically nothing.
First up, you need your DNA template. That's the original strand you want to copy. Next, you need primers. Think of these as little bookmarks that tell the machine exactly where to start and stop copying. Then you've got the engine of the whole operation, an enzyme called DNA polymerase. And of course, you need ink for your photocopier. These are the raw DNA building blocks, A, T, C, and G, also known as nucleotides. And you mix all of that in a buffer, which just makes sure the chemistry is perfect for copying. Now here's the real breakthrough, the secret sauce. The whole process needs a lot of heat, and early on the polymerase enzyme, the copy engine, would get destroyed every single time they cranked up the temperature. It was a huge pain. The game changer was finding Taq polymerase. It's an enzyme that comes from bacteria living in the boiling hot springs of Yellowstone National Park. This little guy loves the heat, which meant, finally, the whole PCR process could be automated. And just like with real photocopiers, you can choose different engines for different jobs. Taq polymerase is your fast, reliable workhorse. It gets the job done quickly, but it can make a few mistakes here and there. For jobs where you need absolute perfection, like sequencing a gene, scientists use a different enzyme, like Plateau polymerase. It's a bit slower, but it has a proofreading ability, so it's incredibly accurate.
So, we've got our ingredients. We've got our heat-proof copy engine. The process itself is actually beautifully simple. It's just a 3-step cycle that repeats over and over. Heat, cool, and copy. That's it. Here's how it works. First, you heat everything up to about 95 degrees Celsius. This denatures the DNA, which is a fancy way of saying it unzips the double helix into 2 single strands. Step 2, you cool it down. This allows those primers we talked about to find their exact spots and lock on. And finally, step 3, you heat it back up just a bit to the perfect temperature for our Taq polymerase to get to work, adding all the building blocks and creating a brand new matching strand of DNA. And that is one cycle complete.
But here's where the magic really happens, in the chain reaction part. See, this cycle doesn't just happen once. It repeats again and again and again, and this is where we see some seriously explosive exponential growth. Let's watch how we get from just one copy to billions.
After just one cycle, our single piece of DNA is now 2 identical copies. A good start, but we're just getting warmed up. So you run the cycle again, those 2 copies become 4, you run it one more time, and the 4 become 8. You can already see how this thing starts to take off, right?
But now, let that cycle run about 30 or 32 times, which, by the way, only takes a couple of hours, and you get this. Over 1 billion copies from that one single starting molecule. That is the jaw-dropping power of PCR. You've taken something totally invisible and made more than enough of it to see, study, and analyze. So it's obviously an amazing tool for the lab. But why should we care? What does it actually mean for us out here in the real world? Well, let's look at the incredible impact this one technique has had on pretty much everything. PCR is, and I'm not exaggerating, everywhere. In forensics, it's the technology behind DNA fingerprinting. In medicine, it's how we diagnose genetic diseases and quickly detect viruses or bacteria. In research, it lets us study DNA from ancient mummies and map out the entire human genome. It even keeps our food and water safe by spotting harmful microbes and helps us protect endangered animals. If a field relies on reading DNA, you can bet it was transformed by PCR.
And this timeline just shows you how fast it all happened. The basic idea was described in 71. The first real-world use was in 85. Less than 10 years after that, it wins a Nobel Prize. And today, it's a standard must-have piece of equipment in basically every single biology lab on the planet.
And that brings us to one last big thought. We now have this incredible power to find and read tiny, ancient, hidden pieces of genetic code from almost anywhere. So the question PCR leaves us with is this: what other secrets about our past, our present, and our future are just sitting out there, waiting to be discovered, hidden in those invisible traces of DNA?